Hi everyone, this is Dr. LeBlanc. This is the first of two video captures for Chapter 14. In the first one we're going to discuss the process of transcription, specifically in prokaryotes, and then the second screen capture will talk about RNA processing in eukaryotes. Right here is a diagram of RNA polymerase, and you notice that the squiggly lines here in gray and yellow are the protein component of the polymerase, which grabs onto DNA, breaks the double helix, and then copies one strand of the DNA into RNA in red. In all the diagrams in this capture, the RNA is going to be red. So a gene is a segment of DNA used to make a functional product. That's mostly correct. Transcription is the process of copying the DNA into RNA. And if you remember the central dogma, when we have DNA and we make more of it, that's called replication. And if we have a gene and we make an RNA copy of it, that is called transcription. Oh my goodness, this is terrible. Transcription. That's what we're going to be concerned about in this chapter. In the next chapter, we'll talk about the process of translation. RNA transcripts have different functions. 90% of the genes that are transcribed into RNA produce functional messenger RNA that gets translated into protein. However, there are other genes as well whose functional product is not a protein but an RNA. Ribosomal RNA, tRNAs, microRNAs, SNO RNAs, which are involved in ribosomal RNA processing, snRNAs, which are involved in mRNA processing, and then there's other RNAs as well. This diagram is one that you should have completely memorized by the end of this chapter. We start with a DNA molecule, and on the DNA molecule we can identify specific sequences that have specific roles in the process of transcription and also in translation. So a gene is defined as a regulatory region, a coding region, and another regulatory region here. The promoter is the region of the DNA to which RNA polymerase will bind, right here. This is where the RNA polymerase is going to bind. Upstream, in this direction, there are other regulatory regions that other accessory proteins will bind to help RNA polymerase bind here. So this is where RNA pol will bind, right here. The terminated sequence we'll get to at the end of this capture. And the process of transcription makes a copy of this region of the DNA and into an RNA molecule. Now embedded in this coding region of the RNA molecule are things like a ribosome binding site, the starts codon for translation. This is where the first amino acid is encoded, right here. Many other codons. Remember that 3 nucleotides equals 1 amino acid. Okay, each codon is a sequence of 3 nucleotides. 3 nucleotides, 3 nucleotides, 3 nucleotides, and each 3 nucleotides equals an amino acid. Okay. Also embedded in this code is the stop codon, where protein synthesis terminates, okay, which is not to be confused with the transcriptional terminator, which is here, which is where RNA polymerase stops making the RNA. So we're going to be concerned with this process for now, and we'll get to the other process later. All polymerization reactions have 3 steps. We saw the same thing for DNA synthesis. we saw that there's an initiation reaction, an elongation reaction, and a termination. We mostly focus on the initiation and the biochemical process of the elongation steps. And we'll also take a closer look at transcriptional termination when we talk about transcription. So here's a gene. Here's our promoter region where RNA polymerase binds, our terminator where RNA polymerase stops its work, and here's RNA polymerase binding right here. We're going to pay attention to the transition from a closed to an open complex. Okay, an open complex is an RNA polymerase that is actively transcribing RNA. Okay. Initiation. RNA polymerase binds at a promoter. Okay, here is an up-close look at a prokaryotic promoter. We're going to look at some of the actual sequence of nucleotides that you would find in a typical promoter for a prokaryotic gene. This is the whole promoter region. This is the region where RNA polymerase binds. Let's orient ourselves a little bit first. Nucleotide number +1 is the transcriptional start site. This is the first nucleotide that is transcribed from DNA into RNA by RNA polymerase. The bases preceding, that means in the upstream region, in this direction, are numbered in a negative direction. There's no base that's numbered 0. Okay, so it goes from +1, the next nucleotide right up here would be -1. Okay. The bases to the right are numbered in a positive direction. So all these plus numbers indicate DNA nucleotides that are going to be transcribed into RNA, right down here. Most of the promoter region is labeled with negative numbers. We have a specific sequence called the -10 sequence. This is an actual specific sequence of nucleotides, T-A-T-A-A-T. Notice, what do you notice about this? Yep, you're right. And we also have a sequence of nucleotides that's called the -35 sequence. Not so AT-rich, like this one is. And in between the -10 and the -35 region is a short segment of approximately 16 to 18 base pairs. It can be any sequence. The most important thing about this region is that it is 16 to 18 base pairs, not too many fewer or not too many more nucleotides in length. Here's a diagram of several different promoter regions from many different genes found in E. coli. There's the lac operon, which we're going to talk a whole lot about after break. After the next exam. The lac-I gene, the trip operon, rec-A, very important, tRNA tyrosine for tyrosine. If we look at all of these and many, many, many more sequences of promoters, we can come up with a consensus. What does a typical RNA polymerase promoter in prokaryotes look like? What's the sequence of nucleotides? Typically, the most common nucleotides we find in each of these positions in the -10 region is T-A-T-A-A-T. And you can look up here and see if you find any that are exactly like it or how often those nucleotides differ. Here's one that's exactly like it, the rec-A gene. And the rest of these vary in different amounts from this consensus sequence. Similar story for the -35 region up here. Okay, let's look at RNA polymerase. In E. coli, the holoenzyme, that means the enzyme with all its subunits attached to it, consists of excuse me, a core enzyme plus sigma factor. The core enzyme consists of, let's see, an alpha subunit, another alpha subunit, a beta subunit, a beta prime subunit, an omega subunit that's behind us that you can't really see, plus a very important factor called sigma factor. Okay. So this is what initially binds to the DNA molecule, and transcription is going to proceed in a left to right direction. One of the interesting things about sigma factor binding to the DNA is that it has a typical motif found in many DNA binding proteins, a helix turn helix motif. This is an alpha helix and then a few amino acids and another alpha helix binding in the major groove of the DNA. Here's a bigger picture. The transition from closed to open complexes is the critical point at which RNA polymerase stops scanning along the DNA and actually initiates RNA transcription. So here's our holoenzyme. Here's our holoenzyme. It has all of the regular subunits, two alphas, the beta, beta prime, and the omega subunit, and it has a sigma factor bound to it. When the holoenzyme is attached to DNA, it's a fairly loose attachment until it reaches the promoter region, where it transitions into a very tight binding at the promoter region. This -10 and the -35 contact different regions of the sigma factor. Okay, so after sliding along the DNA, sigma factor recognizes a promoter. Okay, it's the sigma factor that recognizes the DNA, the -35 and the -10 sites. And our RNA polymerase forms the holoenzyme forms a closed complex that's bound tightly at this region here. Okay. What happens next is critical for the transition from tight binding to elongation phase. Okay. So, So the next thing that happened is two things happen simultaneously. Sigma factor is released. Okay. In order for RNA polymerase to escape the promoter, you need to release sigma factor, okay, and transition from the closed to the open complex. That means that the two strands of DNA are now separated from each other and transcription can begin. So the core enzyme then is able to move down the DNA and make RNA. You have to release sigma factor in order to do that. And that process of releasing sigma factor and proceeding through to the elongation phase is called promoter escape. Okay, here's a little bit more up close and personal about what's happening at the elongation phase. Okay, let's start at the beginning here. So the first thing that happens is that you've transitioned from a closed to an open complex. DNA is unwinding. Okay, here you see the two strands of DNA in purple and blue separating from each other. Nucleoside triphosphates enter in through this pore, and you start to add nucleotides in a similar type of reaction as the DNA polymerase performed when adding a nucleotide, creating new phosphodiester bonds. The DNA behind is rewound, and you keep progressing along the DNA until you reach the terminator. Okay, up here in this box, we're looking at the two different strands of DNA. There's a coding strand and a template strand. Template strand is fairly straightforward. The template is the one to which you bind the new RNA nucleotides by complementary base pairing, G and C, A and T. Okay, the coding strand just kind of hangs out there, but when we look at the coding, the sequence of nucleotides on the coding strand, they are exactly the same as the sequence of nucleotides on the RNA that's being transcribed. So remember, RNA has uracil in place of thymidine, but the C is the same, the A is the same, and the G is the same. Okay, so the coding strand has the same sequence as the RNA does. This is another picture depicting the same idea, maybe perhaps in a little different way so that you can understand it a little better. The coding strand here has the same sequence as the nucleotides of the RNA, A, C, 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 T, U, A, C, etc. All right, so as far as genome organization in prokaryotes, genes are very closely packed together along a chromosome, and individual genes can be transcribed from either strand of the DNA molecule. So if we look at three genes here, gene A, gene B, and gene C, A, B, and C, you notice that A and B are being transcribed in the same direction, but C is not. C is being transcribed in the opposite direction. And that means that the two different strands of DNA act differently for gene C than they do for gene A and B. In genes A and B, the bottom strand here is the template strand, okay, and the top is the coding strand. But in gene C, the template strand is the top strand. So along a given stretch of chromosome, you can have genes transcribed in one direction, in the same direction, or flipped around from the opposite strand and transcribed in the opposite direction. What about termination? There's two different types of termination. There's row-dependent and there's row-independent termination. And that's going to be taking place at this point on our gene. Remember, the gene is the entire segment of DNA, including the promoter region where RNA polymerase binds, the DNA in between that encodes amino acids, and the terminator where RNA polymerase will stop making RNA. Okay, so we're going to look at the row-dependent termination first. In row-dependent termination, there's a terminator here, and there's also a rut sequence encoded in the DNA. Now, the rut sequence does not function until it's transcribed into RNA. Okay? RUT stands for row utilization site. This is actually a Greek letter called rho. It's not a P. Okay? This is the row recognition site in RNA. The rho protein recognizes this RNA. Doesn't bind to the DNA. It only binds to the RNA after it's been transcribed. So the rho protein binds to the RUT site in RNA right here. And it travels then along the RNA towards the 3' end. Here's our rho protein. It looks a little like helicase. It surrounds the strand of RNA. It's a multi-subunit protein. It surrounds the strand of RNA, travels along the RNA, and when it hits RNA polymerase, a stem loop causes RNA polymerase to pause. Do you remember what a stem loop looks like? Okay, here's our stem loop right here. The nucleotides are hydrogen bonded to each other here. There's a loop. Okay, when the stem loop forms, the RNA polymerase then dissociates from the DNA, and everything dissociates. The rho protein comes off, the RNA polymerase comes off, and the RNA molecule itself is released as well. So RNA polymerase pauses due to its interaction with the stem loop structure, right here. This stem loop does funky things to RNA polymerase. The Rho protein catches up to the open complex and everything separates. The DNA-RNA hybrid separates, and termination is complete. Okay, sorry. What about Rho-independent termination? Rho-independent termination is just like Rho-dependent termination. It has a stem loop that causes RNA polymerase to fall off. It just doesn't have the RUT sequence or the Rho factor. The stem loop that causes RNA polymerase to pause forms as the RNA polymerase gets towards the end of the gene. And the sequence of nucleotides here is U-rich. Okay? It's U-rich in the DNA-RNA hybrid right here. And remember that U is only going to have 2 hydrogen bonds with A. So it's a weak bond. While the RNA polymerase pauses, the U-rich sequence is not able to hold the RNA-DNA hybrid together because the bonding is too weak. And termination occurs and everything falls apart. So Rho-independent termination also forms a stem loop, but it also has this U-rich region here. What it does not have is that Rho factor. This is for the next video capture.